you all for joining us today. It's my pleasure to introduce to you Professor Jörn Dunkel from MIT. Jörn is an expert across many different fields, and really excited to have him talk to us today about some intersections with dynamics and biology, maybe some data as well. Yeah, I'll just go ahead and give it away to Jörn and let him get started here.
Yeah. Thank you, Nick, and thank you also for the invitation and for coming to my presentation. Yeah, I will in fact talk most of the time, I think, about interesting data that's provided by our collaborators. And so what you can see here on the title slide are systems that span quite a range of scales, and we treat them essentially using the same methods. And I will go into detail basically about this system throughout. So on the left, you see bacteria or yeast, basically that's really at the micron scale. And on the middle slides, you see C. elegans, which is a roundworm, and then also a jellyfish. And those are systems that we study in the context of neuromechanics. So basically trying to understand how the brain controls shape deformation and locomotion of an organism. And then more recently, also we have been looking at even larger scales. So we have organisms that basically fly distances that span meters. And in that case, that's mosquitoes basically attacking a PhD student in David Hu's lab at Georgia Tech. And I will talk about these systems in detail throughout. But what is important here is really that we have been trying to develop frameworks that can be, with minor tweaks, applied to all of those. And the reason that this works is fundamentally that the data has a similar structure. So it's all time series in some tensorial form that we can reduce and then translate into predictive models. And one of the key goals for us is also to back-end control frameworks that would allow us to make something or use these models to do something useful with them. Good. So if I had to summarize what we are interested in, so we try to build mathematical and computational frameworks to understand, predict, and control the dynamics of complex systems. So traditionally, we have focused quite a bit on biophysical systems, but more recently, we've also branched out and have started looking at political systems. And in the very end, so I'll talk a little bit about this data. And if you find that interesting, so there's a data server that you can access and where you can study things that you may want to know about the US lobbying systems, or you may not want to know. So anyway, good. So all the work that I'll present has been done by an excellent cohort of former and current PhD students and postdocs. So we started out basically to look into model inference with Rohit works on PDE learning, so which was basically built extensively on ideas and techniques that were developed here at UW by Nathan Katz and Steve Brunton's group. And so more recently, we have gone on or moved on to models where we also need stochastic components. And that's also in part driven by a need that comes from the experimental data. So very rarely, basically, we get data that's complete or basically of sufficiently high quality. So you have to include some sort of noise, basically, as part of your modeling pipeline. And yeah, so here on the far right, so George was an undergraduate here and did some master's project, I believe, in Nathan's group, and was subsequently a PhD student, co-advised by me and a colleague, Philippe Rigolet. And today I will focus a lot on work that was done by Alistair, basically, who is the top left. He is now in Chicago at the National Institute of Theory and Modeling in Biology, or so, NITMB, probably butchered the abbreviation. And in particular, also will focus on Alex Cohen's work. Alex was graduated, and also you'll see Chen Yi's face and who's a postdoc, who's been driven the Bayesian inference frameworks and development, basically in my group. And Ashu on the right has also been working on, you see some of his work on neuromechanical models for jellyfish. And Chitya, also on the cell level here, together with Shenyi. What I would also like to emphasize, so a lot of what we do is really curiosity driven, and so we are fortunate to have funding from foundations, private foundations, that give us a lot of flexibility, in particular from Schmidt Sciences, who have been great, and also through individual fellowship from NSF and NSDG that gave students basically the freedom to explore different avenues. And I hope basically these things will persist or maybe will be revived in a few years. So for us, it was really essential to have the flexibility and also take risks within our research. Good. So if I had to place us in this diagram, so where on the vertical axis, so the green one, you have interpretability of models, so which I think traditionally was a goal, say, in physics, or also in applied math, so where you have typically fairly low-dimensional systems, but you try to develop models that at least, I mean, subjectively, we think we understand quite well. Then you would be fairly high up, basically in the top left corner with the traditional theory. And then on the horizontal axis, the axis basically is labeled by complexity, and you can measure that in different ways. You can measure that in basically the data dimension, or also just the size, basically, or the number of degree of freedom, which correlates with dimension, or also maybe in the amount of money that you have to spend. And so you have, these classical machine learning methods that are basically in the bottom right corner mostly. I think, they've become more interpretable, but there are still questions basically of how they function and what the models actually tell us in terms of structure beyond their predictive power. So you have these physics-informed neural nets, basically, that place, say, or pins that place physical constraints. And so we know, at least to an extent also, so what type of priors we built in to these models. And so I think where I would place us is somewhere here. So we are in a regime where we have data that cannot be tackled, I would say, efficiently with traditional methods, but also is not so high dimensional that you could just apply brute force machine learning methods. Maybe you don't want to do that also. And so I think that is maybe where traditionally also dynamical systems learning basically found its origins. And what we do, builds extensively on previous work. So I've highlighted basically the work that was done here, or the groups that have been very active here and pioneered basically recent developments. But so the first attempts, they go actually relatively far back. I think Jim Cessna's group has done some very interesting work early on constraining reaction diffusion systems from data. So if you have heard about sloppy models or so, and the questions that arise basically in those contexts, so they're still relevant to us. And there is now, I think, a fairly active and also growing community, and especially now also with our PhD students and postdocs basically becoming assistant professors, I think that community will grow. And we try to contribute there through methods development. And also for me, it's very interesting. So I have a chance basically to learn about many different areas. So I wouldn't subscribe to the fact that I'm an expert in many areas, but I'm learning about many things really from our collaborators. And fundamentally, if you think about how data is generated in experiments these days, so pretty much all the experimentalists use similar microscopes, for example. So there is a fairly, I would say, limited set of tools that is available to extract information. So it will be video data, it will be some sequencing data, and so on. So you can actually apply, if you set these things up in the right way, to a wide range of systems. Good. So this is basically the structure of the talk, more or less. So I'll spend quite some time to talk about basically the data, and I'll start basically in a second. But my view of basically how we try to model things has been changed quite a bit over the last 7 years or so. So initially, my background was more in the traditional applied math modeling community. So I'm thinking about more ODEs, or ordinary differential equations, or partial differential equations, or deterministic models. And there's the question, so, how would you set these models up so that you can fit them to data structures? And so a typical data structure here, so put into this form, so there'll be a video where one dimension is time and the other dimensions are really space dimensions, or in a political systems, basically it could be, some other axis, could be probability tensors that you can extract. And so I've gotten a bit away basically thinking about deterministic models within the first place, but we really think about now these days as inferring conditional probability distribution. And the reason is that also the deterministic models fall under this umbrella. So in that case, your conditional distribution is simply a delta function in a sense. So, but for most systems that we deal with, you want a stochastic differential equation or some sort of stochastic model anyway. So we think more about basically this. So fundamentally, a model for me is something that allows me to make a prediction about how my system changes. So if X is a variable that describes the state of the system that I think is, the right state space, could be something that's very physically interpretable, or could be something more abstract. And then typically what you want is given basically some information about the history of the system. Ideally, maybe you want to use very little of the history. So that's basically the past. And then also some control variables here indicated by C. So basically, if you knew that distribution, you had a model. And the whole question really just boils down on how you parameterize basically these distributions. And there are many different ways. So you can do it explicitly just by fitting some functions. I'll show you that. Or you can also, learn Fokker-Planck equations or PDEs. So there are many different ways of encoding that type of information. But that's how I like to think about all of these problems right now. And so the last step is, so once you have a model, so what can you do with that? So you can, of course, test it on your validation data set, but maybe you want to do a little bit more. And maybe you want to go back to our experimental collaborators and want to say, if you have these knobs in your system and you have a certain objective, then if you were able to tune these knobs in an optimal way, so you could, for example, steer a worm along a predetermined trajectory. So I'll come back to that problem. I think that's very important because at least my work early on also stopped usually when I had a model and somehow I could draw a curve that fit some data. So I was happy, but I think model actually should give us a little bit more. And we now with, especially with the automatic differentiation methods that are around, we can actually quickly differentiate objective functions through simulations, and we can really back-end optimization and control frameworks. Yeah, so this is the outline, and I will now just talk about basically different data sets to give you a little bit more of an idea about basically the data we're interested in. I mentioned that on the cover or title slide. So these are basically different systems. And what you can do for those systems and what biologists are interested in is their growth behavior. So, I'll start a video here. So that's called a mother machine. So it's a microfluidic device where you have basically a bunch of channels and you try to pack them in a way that, you efficiently can run many of these experiments in parallel. And so what you can see here is these little white bars are actually cells growing in these channels. And so they are fed through the middle, and then they grow out at the bottom and at the top, and then they're flushed away. And just by doing some basically image analysis, you get curves like those at the bottom, so which measure basically the size of a cell basically over time. And so whenever there's a drop down, so it means the cell has divided. And in this way, you get a huge ensemble basically of these trajectories. And what biologists are interested in is really how cells basically decide when to divide. So both basically if they're just sitting there, also, maybe when they're in a tissue and so on. And so there are different ways of, or different models have been proposed. So there are these timer models where you think about cells essentially having a clock, and then once they hit that clock, basically they roughly divide, maybe with some variance, or there are some adder models where cells somehow measure how much mass they've added, and then they will try to divide and so on. And it's not quite clear. I mean, so these models, they sound all plausible, but I think you would need basically quantitative models somehow fit to the data that encapsulate these concepts, and then you would want to make basically a classification basically of different systems. And it's probably not basically very clear that one system just behaves basically as one of these types of systems, but there may be mixed strategies depending on environments and so on. So what we did is using basically ideas that are very similar to what I will describe in more detail later on, basically fit stochastic differential equations models, SDE models, to these type of data. And here the difference from basically standard models was just that we don't have Brownian noise or white noise, or Gaussian basically noise, but in this case, we have Poisson processes that drive these distribution. You try to estimate rates and so on. And then with these function G and H, they encode essentially growth behavior and the division strategies. And given basically the terms in these functions, you can then start to plot these phase diagrams and try to place different systems. And you have your basically standard models that basically are represented by these black dots. And then you can see where actual systems lie. And you can really basically make quantitative statements about these systems. So this is basically all on the micrometer scale. And I will talk a little bit more in detail about basically this system. Later on, it will be my first example to explain how we use spectral representations to lower the dimensionality of these systems. What you can see here is actually an alga. So it's a single cell organism, and it has this bundle of filaments. So actually, I think 4 or so filaments that are braided and form this. And what you can see just by looking at the video, you see quite different behaviors. So what our biologist friends there are interested in is also how many behaviors can such an organism basically exhibit? Because it doesn't have a brain or anything. It's just basically moving around. And so also then what determines eventually the transitions between these behaviors, how long it is in a certain behavior and so on. But so for them, it's a challenge to quantify these things systematically, and I'll show you basically how you can use spectral representation to do that quite efficiently. So that's also on the micrometer scale. I will spend the main part of the talk talking about these systems, and those are these neuromechanical model systems where there has been immense progress over the last decade or so in developing experimental systems. So C. elegans on the top, so this worm, where you can see basically individual neurons light up when they are active. And I'll show you basically what these curves will look like. And simultaneously, so our collaborators in Steve Lavell's lab in Brain and Cognitive Sciences at MIT have been developing a microscope where they can track the motion of the organism and also observe part of its brain, basically, as it fires. So you have a complete data set, modulo some measurement errors and other problems, basically, that always arise when you work with real data, where you see both the neural activity and the actual physical behavior of the organism. And so the question is to obtain models that would, for example, be able to take the neural signal as an input and predict the locomotion behavior. At the bottom is more or less a two-dimensional version of that, so which is a jellyfish here basically confined between two plates, and that's a very recently developed experimental system by Brady Weisburg, actually at Caltech, basically at that time, so he's now faculty at biology. And here you have many more neurons, but I'll show you later some videos basically how neuron activity leads to deformations of the body, and we try to develop models that connect these different layers. So that's a typical example of a multi-scale basically data set where we have, say, some biological component, which is a neuron activity, and then we have more kind of a continuum response of the whole organism to this localized activity. So these organisms live roughly on the millimeter scale, so jellyfish here are quite small in that case. You can also go to much larger scale. So this is work that we actually tackled using more these PDE methods. So from Ian Cousin's lab. So these are fish swimming in a tank, and you can track them, and then you can try to infer models about pair-based interactions. So what is the strategy basically to follow them? How do they respond to external signals? So in that experiment, for example, so the fish received an external stimulus, which was a light pattern that was rotated, and it acted almost as an electric field or kind of magnetic field and made them align. And that's on the meter scale. This is also on the same scale. Basically, you can ask, can models that you infer from data help us to understand how insects search basically their targets, how they attack them, what do they respond to really, how do they combine stimuli? And if you go, so here's maybe just to give you a little bit more of an idea. So these experiments were performed by David Hu at the CDC, and this is basically the mosquito type that they were using. And this is Chris, basically, again. So Chris was, when I always get this data, I'm happy that I'm just a theoretician or the guy who works with the data and don't have to bother about getting them. And in that case, so we got a really groundbreaking set of trajectory data. So they used 3 cameras to reconstruct the trajectories of individual mosquitoes. And in that case, we were able to infer stochastic differential equations, essentially a bit like Newton's equation of motions for mosquitoes, that tell us basically how certain type of target stimuli. So, for example, the color of piece of fabric that you want maybe wearing, or CO2 concentration, or also the heat, basically that your body emits, are seen by the mosquito as an effective force field, basically that steers them, basically, and guides their response. So it was funny. So David wrote the paper came out quite recently, and then David was asked to put basically a public, the accessible piece, and so he basically recalled how his interaction with Chris went, and Chris told him that 4 minutes is definitely too long, so he has to be in there. So you get too many things. Good. So this is all on the meter scale, and then on the kilometer scale, it's here basically an entirely new data set, and I'll talk, if I have some time at the end for 5 minutes or so, I'll talk about the data set in more detail. So this is a data set that encompasses the complete federal lobbying history of the US since 2000, roughly. I can give you more details. So there are interesting questions, basically. You can ask, what is a successful lobbying strategy once you have quantitative models for that? And, of course, we don't know really. So I'll tell you basically a little bit how this data is collected. And there are certainly reporting errors, and people probably are not entirely honest, basically, about how they do these things, but at least there's some data. Good. So when you deal with the system across various scales, so you typically encounter the very similar challenges. So all these data sets, so they are large, but they're still finite if you think compared to LLM, also that use these really internet scale data sets. So one of the reasons that the data is finite there is that it's quite expensive to collect those. So, for example, to build these microscopes that can measure neuron activity and also the locomotion. So that takes a lot of time, and you need PhD students to do that. And so it's very unlikely that we will get a lot more data there. So you really have to think more about the structure of the models rather than just feeding that data into an LLM and, type in some prompts and get the answers that you may want. So another limitation, especially when you work with biological systems, is also that you have limited spatial and temporal resolution. And sometimes it's just a limitation of the microscope, that you have a certain frame rate and a certain resolution if you want to look at a certain, basically, field of view. But then you have to compromise with your spatial resolution. But sometimes it's also just the fact that you may be dealing with biological systems. So you typically, when you image them, you have to excite them with a laser because they're fluorescent signals, and they don't like that. So if you heat them up all the time, so they will not behave like they actually behave. So in that case, so the spectral representation that I'll be talking about can help you to interpolate the data and basically fill in some gaps under some structural assumptions again. Yeah. So another aspect is often that the observational basis is, I think in most cases, not optimal if you want to write down models. How we perceive our receptors or eyes, for example, how processes happen, and the microscopes essentially mimic that. So that's typically not these are not the coordinates in which you want to describe your system. So so you have to think a little bit about basically how you can do dimensionality reduction, and there are many different ways of doing that. So I like the ones that are essentially linear transformations, where I know what I do, but it's just a matter of preference. You can also use more abstract methods. I think another important challenge is that in many cases, the data that you have is incomplete. So say for the neurons, we can observe only typically an experiment for C. elegans 150 out of 300. And so you're losing some information. And now what you can try to do is to take all these missing degrees of freedom and put them into some noise. But these are challenges that almost always arise that I don't know of any system, except maybe if you observe a pendulum or so, that you that you have a full complete information. Then there is basically other things that are typical, like measurement noise, tracking errors, and so on, and non-Gaussian noise. Good. Okay. Good. So let me now go a little bit more in detail into one specific example to talk about spectral representation and how they can give you low-dimensional representation that can help in biology. So if we think about spectral representation, then really what we do is we have basically a tensor, basically of some sorts, or that stores our data, and we want to try to reduce the dimensionality by applying some linear transformation. So you can think of these phi matrices here being Fourier transformations, but typically we take basis systems that somehow are tailored to the type of geometry that we consider, and so on. And then you want to typically work with that reduced variable tensor C in the center, and this is the space in which you basically formulate your models, and then you can reconstruct your video basically from your model if you wanted to. So, and these spectral representations, so they typically help us. So what you can do, you can get a data set. For example, you can apply different candidate basis systems, so different phis basically, and see what gives you the best compression. And if you find one that is much better than others, so you have already learned something about your system, because very often the spectral basis reflect some underlying symmetries, for example. So they also give you nice interpolation properties. If you want to differentiate data, I mean, we've moved away from that, but if you wanted to, so it's also nice once you have the spectral representation, because your derivatives just act on the basis functions and not on the actual data anymore, and so on. So there are some benefits to working with those. So here's again basically that example of pterosperma, which is actually an alga. Movie is heavily slowed down, and the way we get these data is typically by a lot of basically sample points that experimentalists track, or we track ourselves. And if we say have 100 sample points, and traditionally what people have been trying to do for this type of system is model these filaments as basically curves in space. And so you have essentially an infinite dimensional system. But if you look at the dynamics that you see in these systems, so it's fairly low dimensional. So you can actually hope that by projecting on a bunch of modes, on this case, maybe Chebyshev polynomials, so you can pretty much recover 95, 99% of these dynamics basically in a much reduced space. And it is illustrated here. So what we will look at also later on for the worms is a curvature coordinate is essentially the tangent angle, basically defined to some reference. And so you have that angle basically at each point of the filament at a given point in space, and alpha is arc length parameter. The parameter is the position where you are relative basically on the organism. And you have a field essentially in T and alpha, and you can expand here along the alpha direction in these modes. And what you end up with are these mode coefficients that encode basically the curvature basically. So the way these are defined is actually, so the blue one, so theta 1 would correspond to a simple bending mode, and then theta 2 basically to higher order bending modes. And that's because it's already an angle and so on. So, and it turns out, so for example, for that system, so if you just take 10 modes, you pretty much recover basically all the dynamics. So there's no need to try to model basically in real space, basically curves and so on. Now, a nice thing that happens also when you look at the dynamics of these different modes, so these theta coefficients, so you see that just by inspecting that they strongly reflect behavioral transitions. So in time, so you have a different type of oscillations basically that the organism performs, and they are nicely reflected in this lower dimensional space. And so what we typically do for many systems now is these wavelet transforms or windowed Fourier transforms in some sense, which give you an instantaneous frequency decomposition. So here are these different mode amplitude curves for these various modes. And what you can see very nicely that initially all the energy is localized at a certain frequency, and then there's a transition, and then you enter different behavior, and you see it very nicely in that frequency space. So here's again basically how it looks in time, and now as you follow that vertical curve, so you can see that once you hit basically where the point where the spectra ends, so you'll see that the behavior also changes quite dramatically. So now so it stalls and does something else and goes to a different waveform. So what I show at the bottom is really just, so if you treat with these columns, basically of these stacked matrices as vectors, and you look at a low-dimensional representation, so you see that you move basically in that abstract spaces, or like a PCA space, basically between different behaviors. And so if you measure basically the distances also, so you see that this gives you a nice measure just of clustering, basically with different behaviors. And so that was addressing one of the questions that our collaborators had. So, how many different states can we actually distinguish, and, how do they differ in terms of their physical characteristics and so on. Can I ask, is actually a physicist here? Somebody who has a physics background? No? No? Good. Then maybe you can't appreciate that slide so much, but so what you can see here on the right and next to are these different modes, so the bending modes essentially, and they correspond to different spatial wavelengths of your system. So here you have frequency on that axis, and you have time on this axis. So you have essentially, if you stack the matrices behind, you have a three-dimensional tensor. So now if you integrate sum over the time dimension, you get the frequency as a function of the wavelengths, effectively. So this is typically what you would get to know or learn in a physics course as a dispersion relation. So if you solve the wave equation and so on, but here you extract it directly from the data. So in essence, you have some sort of temporal decomposition of your dynamics, you have some sort of spatial decomposition, and you plot the magnitude of the coefficient, the expansion coefficients in these plots. And now what is nice is that you can see how different behaviors correspond to this dispersion relation. And for physicists also, if you want to extract these things for a crystal, so that's, how physicists, condensed matter physicists, a lot of them basically made their money for a long time to extract these structures or band structures, you would typically have to excite the system from the outside, and then you would measure the response. And if you see a response basically at a certain wavelength, so you draw a dot and so on. So here, because these systems are alive, so they explore a part of the dispersion relation automatically, so they have excited. So you can extract this dispersion relation, and during its life cycle, the organism will jump basically on these branches and perform different waveforms. And you can actually back out basically what these different waveforms are. That's on the right, and how they excite different parts of the spectrum. And then behavioral transition actually correspond to jumps, basically, or transitions in these dispersion spaces. You can also just do basically something similar to refer to these, just look at here, essentially PCA, principal component embeddings, and you get something like a behavioral manifold, and you can actually trace the dynamics basically of the organism on that manifold and see how it transitions. And what our collaborators try to get out of that is also to understand if there are certain cycles, if there's certain memory, so that you don't jump randomly between behaviors, but maybe if you've been in one, you go into the other and so on. So it's like, if you had dinner, then you probably go to bed and sleep, and then you wake up in the morning. So anyway. Okay, so now I want to slowly transition to more basically questions that are of interest to us. So how would we try to learn dynamical models? I will not illustrate it for that system because we have a more interesting system, which are the worms, but the idea is the same. So I can introduce the data structure here already. So if you have parameterized your filaments basically in these ways, so you can plot the pairwise phase diagrams of these mode coefficients. So those are now our variables for which we wanted to learn dynamical systems. And you can see if the organism is in one mode, basically where it pretty much swims straight. So you get these phase portraits that have a certain shape. You see some that have basically very simple structure, and then some oscillations, basically where you just flow in these coordinates and so on. So but it happens in this 10-dimensional space. And if you went to basically another state where it turns, you see that these change quite a bit. So that means that if you set up models, so you have to find a way of also either, the idea was now to have a model that automatically transitions between all these different states, or you have to build hybrid models. And that's what we are currently doing, is where you identify a state and then you identify the time basically the organism lives in that state and the probability it jumps to a next state. And then you learn different models basically for these different states, and you can compare all the states on those models. Good. So we take these same ideas and apply them to many different systems. So for the worms, basically we use this pretty much the same, and I'll talk about in detail about this. Jellyfish, you take different basis expansions. So For example, Fourier-Jacobi is really nice to, in terms of numerical properties. You can also do it on spherical surfaces. I think Nico Romeo will be visiting here, so that was his work. So he was looking at the migration of cells on essentially a sphere, a yolk ball in a zebrafish during development. And there, these type of vector spherical harmonics and scalar spherical harmonics basically are very convenient. Good. So now let's briefly talk about model inference. And what I do is I go at a very high level through a toy example just to highlight the same ideas. So in the end, we do pretty much the same for a more complicated system. It's a bit more tedious then, and you have to do a bit more fine tuning, but the ideas are the same. So what I did here is I generated for me a test data set, which is comes from this one-dimensional toy example. And just to give you an idea. So here it's a very simple system. So you have basically an overdamped dynamic. So your spatial increment, if you divide by dt, you get your normal differential equation, stochastic differential equation. So here it's written in the form that mathematicians would prefer, where basically the increment is entirely determined by the derivative of a potential. The potential here is of 6th order. So it has 3 minima actually. And then there is a noise term at the end, which gives you some basically accounts for degrees of freedoms that you don't explicitly model. And they usually also assume that the dynamics happens on a much faster time scale than for this degree of freedom. So if I take that as basically a model and I just integrate it forward in time, I get a time series for x and t, and I can do that for many different realizations. And now I can treat that data set, so which is just these x of t's here, as my just my experimental data for a moment to illustrate. And in a real experiment, we get that type of data, for example, the mode coefficients from the experiment. Now, what you can do is you can plot just the dx, so increments that you just take the difference between successive points and you plot them against x. So here it's a nice system. And what you see, effectively, you have this joint probability distribution in the second slide. And you see that it's peaked basically where the minima of the potential are. And what I've shown in red is basically a curve that you get when you just fit basically a polynomial of a certain type, basically through that point cloud. And this is essentially what we do basically whenever we fit a model. So at some level, we do manifold fitting basically to a point cloud in that increment space, or where basically on one dimension is the increment, on the other dimensions are your state variables. So you can also see what would go wrong. So for example, if I didn't have enough data, so I wouldn't see the minimum on the left, I would probably get a model that is not quite accurate in that regime. The other thing that is also interesting is now you can, once you have inferred basically that red line, you can transform your data. So essentially you solve your equation for dB, and you know that dB is white noise, so you would expect a Gaussian. So if you have basically start from a modeling assumption that your conditional probability distribution is generated by a certain noise structure, then after basically if you have done your fit, you scale back, you should recover that noise structure. So those are cross checks that you can do. So here, that's of course a toy example. So we have a completely determined system in the sense that I can, knowing x, basically I can predict the statistics of dx. So now in reality, that's never really the case. So here's an example that just illustrates what happens if you say observe just dx, because your experimental setup only allows you to observe dx, but the system actually depends on 2 or more state variables. So here I took 2 variables that are coupled. And if you just plot dx against x, so you see that you now have these 3 almost deterministic curves or manifolds basically that live in parallel there. And so that would be a signature of just having not enough observational data. And then you have to think about things that you may be able to do. So maybe you have to think about including derivatives, or you have to change your experimental setup and so on. And the reason is simply, for a dynamical system, basically for every dx, we have a well-defined dynamical system, there should be, so every state should exactly define basically 1 dx, because otherwise your mathematical theory of differential equations would not collapse. So if you now were able to measure both x and y and dx, you see that for every dx now, basically you have a unique, for every x and y state, you have a unique dx, basically in that case. So, and you can estimate the dimensions of your system by doing some target type embedding, delay embeddings, and so on. And so it's effectively the same thing. Yeah. So now, again, basically to find the dynamic system, you just fit a manifold and then you do your noise estimates in that case. And that's just summarized here. So fundamentally, you set up models that are of this type. So it's just written in a heuristic, basically a schematic way here. And if you make an assumption about your noise, and there are also choices that you can make basically when you model or set up your modeling framework. So if your noise is nice, or you may with Gaussians at the end, and then if you do maximum likelihood estimation, you have nice formulas to efficiently estimate the parameters basically of your function f on g that you want to find, and so on. And every system that you can reduce to an SDE pretty much falls into this class. What may differ is what your noise distribution is. So for example, for the cell division example, we had a Poisson process and not a Gaussian process. Okay, so now let's see how that works in practice. For I think one of the most exciting data sets, basically, I had the chance to work with in the last few years. So that's work by Alex Kohn. And this is, again, C. elegans. And here you can see the worm. And on the right, so this is not exactly correlated, but just to give you an idea of the data, you see neurons blinking. So Alex put some stars on them just to make them shiny. But that's the type of data that you get. So typically you get a video basically where you see the midline, and then or the worm basically moving like this, and then you see the neurons. And there goes a lot of work into identifying the neurons. And so the elegans is nice as a model system because it has a very small brain. It has only 302 neurons, but it won at least 4 Nobel Prizes over the last couple of decades in various disciplines. If you're interested in that type of data, so Stiefler Welslab, so they have set up a database where you can download data sets of this type if you want to work with these data. So it's publicly available. Call it the Worm Wide Web. Good. So here is the same data on the right, just different, represented in slightly different form. So we go to a co-moving frame. So we see basically the shape changes of the worm. And then we see 4 selected neurons that are represented in that network. So the network actually shows more of these neurons blinking, and we should just 4 prominent ones. So we worked quite a bit on trying to find low-dimensional representation for the brain. We didn't succeed. So that may be something just related to how we tried it, or inherently there is no low-dimensional representation, or not sure. So in the end, so what we do is we focus on prominent neurons that have basically a strong correlation also with motion changes and so on, and try to set up models for that. So what you can also see there is in that network, so it's about, say, 100 nodes or so, 100 to 150. It's typically what we get for these experiments. And there are these connections, and those are physical connections. So some really tough PhD students or group of PhD students in biology, so they mapped out basically under a microscope how neurons are connected, and it's very stereotypical. There are many other ways for neurons to communicate, and that's, I think, what makes it hard. So neurons can secrete chemicals, and you have a chemical connectome and so on. So it's not quite clear basically how these interactions work. Good. So what we do is, so we want to build a pipeline basically that takes the neurons signal activity, the network activity as an input, predicts the shape changes. Once we have the shape dynamics similar to basically what I showed for the alga before, we can translate shape deformations using resistive force theory into actual locomotion pattern. So that's the easy step to go to basically the second blue arrow. And then we can build our back-end control framework. Now I'll show you results for that. But the key point initially is that we can take these systems basically and frame them, put them exactly into that joint or conditional probability framework as before. So in essence, what you want to do in an ideal world scenario is measure neural activity. You want to measure postures. You have to somehow parameterize them. Postures are basically the shape of the body, and behavior. So, and then there's a question of how you define behaviors, but you can do it in the same way as we showed it for the alga, for example. So if you had access to that distribution, which in practice you don't have for various reasons, full distribution, so you could answer biological questions. So you would be able to make a prediction, say, given a certain behavior, say worm moving forward or worm moving backward, what are typical body shapes or body sequences that would go through. You can also compute very easily, basically, given a certain neural activity, what is a posture, the typical posture and so on. Or you could also invert the whole thing. You could ask, if I observe a certain posture or a certain sequence, can I actually infer or make a prediction about what the worm was thinking at that moment? So if you had the data. So the problem is that you have, so that's the easy part. So the problem is that you have different amount of data for different types. So the video data for worm moving is very easy to get. Neuron data is very hard to get. So now this is where you have to start basically putting structure into your models and into your parameterization of these distributions. So the first step is still basically accurate or exact. And the reason why we split up the probability distribution in this way is that we get a lot of data for the blue part. This comes from videos. It's harder to get data for the first part. So we have to put in more factorization assumptions, and then we have to see how far we get basically by factorizing the model. So we reduce effectively the number of parameters so that we can still constrain them. And then in the end, we have to see if the model is still predictive. Good. So now I will talk you now in 2.5 minutes through 5 years of work basically by Alex. So can't do him justice, but it puts various pieces together. And I think there's also some nice math in how you parameterize the dynamics of these systems. I'll get to that in a second. So the first step is simply what we've seen before for the other organisms. So we take the shapes and we put them into some spectral basis. So here we took the Jörn polynomials, and you can see that there are different basically characteristic patterns for these curves. So from these patterns, you can also correlate. So there is a certain phase behavior between these curves. If basically the worm is moving forward versus was moving backward or turning left, turning right. So those are the states that we consider here. And at the bottom, you see basically your dynamical system basically in one of these states. So if you want to model such behavior, at the moment, we don't have a model that would automatically switch basically just dynamically between these different behaviors. Like say my dream is always to have a Lorentz worm. So where you have a dynamical model and it spends some time basically in one lobe and then in another end here, but we are not there yet. So we have to do something intermediate. So we learn different dynamical systems for these different segments and then learn something about the transition dynamics basically between those. And here are rates. So there's actually also a very faint error between the right over the dorsal turn, so one is right, and so the yellow and the reversal dynamics. You can see that. Good. So what you can learn, basically once you have extracted states from that, you can learn probability distributions for the neural joint probability distribution between these states and these neural dynamics. So the key thing here is that we found that it's not sufficient just to look at the neural traces. You actually have to also include information about the time change of these neural traces. Otherwise, you don't have enough information in there. So it's important to encode the neural, also the change of the neural activity as part of your probability distribution. Good. So now we have that probability distribution learned from data. Now we want to learn the dynamical system. And so if you think about basically the structure of these dynamics, so we want to basically for that project, we took basically stochastic differential equations that were of the type that I showed before. So you have some deterministic part, which is encoded by F, and then you have a noise bit in the end. And that sigma matrix encapsulates your noise amplitudes, and you learn that also from the data. The interesting bit here comes into how you set up the inference for F, for your interactions between the modes. And if you look at the phase space, for example, of this one here, so what you see is that your dynamics somehow converges to this limit cycle, and then it will also basically flow along this limit cycle. And so a natural way that is very also numerically advantageous is to split basically your function f generically, you can do that on a very mild assumption into basically a gradient part and a curl part. So it's a Helmholtz decomposition in that case. So the gradient part basically of that potential is essentially what drives you onto the attractor, onto that limit cycle. And the curl part tells you how you flow around that. And there's something neat about basically how you can parameterize it. So if you parameterize also your potential in a way that that is perpendicular to your curl part everywhere, so the gradient of the potential is perpendicular to your curl part, is an extra condition, then you can show that the probability distribution that you would get basically would be just Boltzmann. So you can just use score function methods and so on to estimate basically your potential part very easily. So now the second interesting part is, or how do you parameterize your curl components, or the C component of your vector field? And what we use there is an idea that is a little bit older in physics. It's called basically Nambo-Hamiltonian dynamics. So who has heard about Hamiltonian dynamics at some point? Who has heard about Nambo-Hamiltonian? No, okay. So I learned also, I basically also just somebody introduced me to that 4 years ago or so, and I was a bit mad and sad and everything at the same time, because I only learned Hamiltonian dynamics, where you're given a single Hamiltonian, and that Hamiltonian is the energy function of the system and describes the dynamics. And so there's basically one conserved variable, and if you have, you may have others in the end. But there's a generalization, and the other thing is also it works for even numbers of degrees of freedom, because you have basically a momentum and a position variable and so on. But there's a generalization to that. It goes back to Nambu. And so they developed that in the 70s, and also some people actually before him, but it is mostly named after him, where you can have n Hamiltonians, and the dynamics conserves all of those. Basically, if you set it up in the way it's written there, where you combine basically these partial derivatives, and it gives you a really nice interpretation of what's going on in terms of geometry in your space. And I will just briefly, if you haven't seen it before, just illustrate it here for the Lorentz system. So you probably all have seen the Lorentz system here, so the chaotic system. So it's a three-dimensional system. And I mean, I never really had a good intuition, so why the equations basically would give me that shape, but the Nambu-Hamiltonian dynamics gives you a very, very nice, basically, geometric insight there. So what you can do is you can write the Lorentz system basically here in this form. So that's not our work, as it goes back to the references at the bottom, but it's a nice pedagogical example. So if you write this dynamical system basically as a vector field F on the right-hand side, you can decompose it into a gradient part and basically a curl part. So the gradient part is simply the derivative of a quadratic potential, which just drives you back always to the origin. It's just a damping that maintains you from going away. So the curl bit you can write in this form as a combination of derivatives of two Hamiltonian functions. And you can work out what these Hamiltonian functions are. So the curl bit C is simply what you get if you subtract the gradient part from the right-hand side, there's something left. And then you can find these functions H1 and H2. They describe those. And these functions, so one describes a cylinder, and the second one just describes basically a parabolic sheet. And if you didn't have the gradient part, your dynamics would be basically of Nambu-Hamiltonian type. It would conserve both of these quantities. Just like in normal Hamiltonian dynamics, you conserve basically initial energy. So what you have basically is an intersection between these two surfaces, but now you have the gradient part basically that wants to push you off these surfaces. And that determines essentially the geometry. So for us, that's now basically how we often approach modeling these dynamical systems. So we first basically split them into these gradient and curl-free parts. So the gradient part, we can just basically estimate like a score function, basically methods or so, just we get the potential. And then we get the geometry of the curl part basically that way. And you immediately have a geometric interpretation basically of what your dynamics does. Good. So yeah, putting all this together, now we can work towards controlling the mind of worms. So first, what I'll do is I show basically how we can use basically models by fitting these type of models to data, basically to recover data or predict data that we haven't seen. So the way it works in practice, so we have learned basically the joint probability distribution, we have parameterized it. So we can now give them basically neural traces we can predict basically the probability of being in a certain state, moving forward, moving backward, turning right, turning left, so on. And what you can show here is, or what you show here is the predicted state version. This is the experimentally observed state. It's not perfect, but it's basically reasonably strong in terms of correlation. So now having that distribution, we can predict basically what the mode dynamics is in that given state. And we can translate that mode coefficient picture into different postures at different time. And then we can again compare with experiments. And you see basically by eye, so it doesn't look very basically bad. I mean, I don't want to bias you, but we were quite pleased. So this is now if you run a video. So it's a stochastic differential equation. So it's a bit more jittery. So we can probably improve upon that. But I think qualitatively, actually, it looks quite good. Now, what you can do is you have a model of this type. You have the parameters in your function. So you can now define a loss function that where you say, well, how would I have to control the neuron traces so that I follow a prescribed trajectory just by differentiating basically through your model. So define an objective, basically a segment, a line segment of, say, a letter, and say, I want to have my organism move along this letter, differentiate through, and basically get the control signal. So you can do that, and Alex did that. And so you can get the worms to spell out basically letters. So here, basically, that's a prediction now of the model. So our collaborators, I hope they will be able to do that within the next few years. So right now, they're able to optogenetically manipulate, say, one neuron. So the difficulty is you would need 4 lasers of different colors to activate different neurons. And you would have to do it in a way that the spectra don't overlap, or the intensity of the laser. So that's quite hard. But in principle, so that just illustrates that once you have a model, you can now go towards basically control problems with these models. And this is, I think, where the strengths of such quantitative models lie. Good. Okay, so let me just basically conclude with a few basically outlook slides where I show some systems we work on right now. So this is a jellyfish system. So here the jellyfish is mentioned. So the imaging is done in Brady Weisburt slug. That's Brady there. And what you can see is that the jellyfish is contracting. And I will show on the next slide basically how you can identify neurons basically that stimulate these type of contractions. So on the right, we overlaid basically a grid. There's a lot of work basically that goes into that. It looks quite easy in the end, but it's there's some. That was actually the most tricky part for that project, just to get good tracking. And that grid allows you to measure spatial deformation, so the elastic response or viscoelastic response of the jellyfish. And you can again Helmholtz decompose basically this deformation field into a gradient part and into a curl part. That's a quite natural parameterization. So what I've shown here is now a couple of neurons that are circled. Actually, so the bright points, if you wonder, on the side, so those are the tentacles of the jellyfish. So that grab also food. And in the middle, so the little diamond shape bit is the mouth. And so these little circles, so they show basically neurons. And what you can see there is the neuron firing patterns basically of those. And you can do that for many neurons basically in it. And then you can see how basically deformation fields in this Helmholtz decomposition. So on the left, I think is only the gradient part. On the right is the curl part. Basically how neuron firing affects basically these different components. And now we try to do the same as for the worm. So we try to learn a model that allows us to predict shape deformations given the neuron input. And then at some point they can probably take, like Star Wars, a laser and fire at certain neurons, activate them, and then steer the jellyfish. Also. Good. And as a last example, so this is now the political lobbying data. So here, I mean, the data type or origin is totally different, but the framework is exactly the same. So I don't know how much you know about political lobbying. I didn't know anything when I got involved in that project. And so the data was database was assembled by In-Song Kim, who is a colleague in political science. So we actually met in daycare. So that's where half of my collaborations actually started. And so we are starting to talk about our work, and he said, oh, we have this data set where we basically pulled all publicly available lobbying information from the internet and curated the database. So in 1995, so there basically was Congress passed this Lobbying Disclosure Act, where it says that if you do federal lobbying as a company or individual, you have to report your lobbying activity quarterly on a piece of paper, and you have to state certain things. You have to state basically how much money you spend, what law firm you talk to. The law firm has to enter which lawyer worked on that, who they talked to, and so on. So all this data basically curated and disambiguated basically is in this LobbyView database. So you can just go there, get a login, and then explore the lobbying history data. So there's about 1 million records in it, and the total money that went in is larger than $100 billion. So the economists, I think, or the political scientists, they argue whether that's a lot or not. Because what you get in return, basically, if you're Apple or Google or so, and you lobby on the right thing, can vastly exceed basically that. So the way basically you can analyze these data, or one way of looking at it is basically to represent as these lobbying networks. So this is just a single year. So we have basically every year. So initially, so there was a ramp up time. So from 1995 to 2000, so you clearly see there are more reports coming in. So it's not steady state, but then basically it kind of levels out. So you have different layers in that network. So you have clients on the left. So here the blob size tells you how much money they spend on lobbying. So you can probably guess it's probably, I don't know, Apple, Google, Tesla, or whatever, these big things. The middle layer are the registrants. So these are lobbying firms. And what I didn't know about lobbying, so there is a street in DC, which is called K Street, where many of the lobbying firms are based. And so either typically you go through that basically path, or you go in-house. So if you're a big company, you probably have a bunch of lawyers that just specialize in lobbying and talking to the right people. So then these lobbying firms, they employ lobbyists. These talk to government agencies, and so they sponsor basically. And then so these government agencies, so they consist of politicians, or they're interns of politicians or whatever. So there are all sorts of connections. So you can map them to members of the House of Representatives or the Senate, and you can also see how they voted in the end. So what you don't know from the report is whether somebody lobbied for or against a bill. You just have to state that you lobbied on a bill. Yeah, and now from these you can again extract all sorts of joint probability distribution, also in a time-dependent way. And you can typically what you have to do is you have to first cluster, because the amount of data is big, but it's not infinite. So to estimate these probability distribution, you have to reduce dimensionality basically here by clustering. And then you can reconstruct basically pretty much whatever you want. So you can find the most likely lobbying pathways conditioned on a topic, conditioned on a certain politician, conditioned on a certain industry, and so on. And you can do that also in a time-dependent way. You can see how it changed during years. You can see how COVID or the credit crunch affected basically lobbying patterns, how presidencies affect lobbying patterns, and so on. Good. If you want to read more about it, so there's a preprint basically, and that work on our site was led by Carol Bacek, who is an instructor at MIT. Good. Yeah, so let me just summarize here. So what this kind of, I don't want to say it's my favorite slide, but it's a slide that I like, because what I show here with these colors is where we are on different systems. So everything that's green is where we kind of succeeded using the methods that I described to infer quantitative models. So the green one are published, the turquoise one, so they are preprints where we think it worked, but the referees have to agree. And then there are basically two other systems where we still basically in progress. So the jellyfish system, I'm pretty optimistic that we get it to work. There's also this red one, which actually started everything, and we still haven't gotten around to actually get it to work. So this is a system where collaborators of ours can measure spatiotemporal gene expression and physical properties of bacteria. So they have it's a really cool experiment. So they have bacterial swarms. So they put a few bacteria in a petri dish and watch them over hours, basically multiply and expand. And so they go in with a microscope and they measure how they move, how they grow, and what not. And then they go in with a pipette after they've taken a video, take a sample out and measure the gene content or the protein content, so on. So you have these spatial temporal protein patterns, and so that was the start of everything, but we haven't gotten it to work. So then we got to other system and simpler system. Yeah. Good. Yeah, this is what I wanted to present here, and I'm happy to take questions.
So we'll start if there are any questions in the room, and then we'll move over to the questions on Zoom. Are there any questions here? Okay.
Really interesting applications here. I wanted to ask if you consider the collective behavior, like in the mosquito and the following.
So for mosquitoes, we didn't look at collective behavior. So that was initially something that we wanted to try, but then there wasn't a strong signal for that at all. So, and I think if there's a target there, then it's dominated basically by just them wanting to go to the target. So you would have to change the setup there. I mean, we definitely would want to do that. So there are other systems where we're interested in interaction. So I didn't actually here, it's on that slide. So that, yeah, it's a bit simpler. It's a two-dimensional system. So these are also the embryos of starfish. And during a certain stage of their development, they look like little toys, and they have little tiny hair in the feet and swim and spin. And they form these crystals, basically, and that is based on interactions. And these crystals persist for a couple of hours, and then they dissolve, and you can form mixtures as normal. And it's really hard to understand the interactions between those. So we apply, in that case, we have been trying to apply these inference methods. So worked to an extent. It turns out that to really get a predictive model, I think we need accurate tracking of the rotation of the spinning, which is experimentally difficult. So it's not just a distance-based interaction, but also has to take into account. But for the mosquitoes, so far not. So that we only focus basically on how they would affect the target. So there are other interesting follow-up projects now. So it's also something I didn't know. So depending on whether I think the victim basically carries malaria or not, so they behave differently, and also the mosquito flies differently if it has blood carrying a malaria virus. So we are trying to work on data and try to quantify in terms of models what changes in their flight behavior based on these parameters, but interactions, so we didn't have data that was strong enough.
Other questions in the room? On Zoom, there's a question. The Nambu Hamiltonian decomposition requires strong assumptions on the form of the potential to specify the curl. In the example you showed, you assumed a quadratic potential. Do you have the general ideas for how to do this in a physically principled way across different systems?
No, I mean for potentials, I mean fundamentally. So for the potential part, I think the potential part is actually quite easy on that form. So what you could do is you could always start just by taking. I mean, as you compute a score function, just it takes a log of your probability distribution in space. So you can always do that. Then you parameterize the fit in whatever way you want. If you then want to be consistent, or at least partly consistent, you have to parameterize your curl field basically in a way that is roughly orthogonal. But what we found is also, I mean, in practice, it doesn't really matter so much, even if you don't enforce. So they are, in many cases, at least the system you look at, they are roughly orthogonal, just good enough. So you get a model that's better than, it depends, I mean, also on the level of accuracy that you want to have. So for many of the systems that we work on, so at the moment we are happy if we get a model that's kind of semi-quantitative predictive, and it's better than everything we had before because we didn't have any model. So the bar is relatively low. But yeah, I think the score function, I mean, or the potential is always the easy part. The curl part is more interesting.
Maybe one last question. All right, if not, we'll go ahead and take our speaker again.
Can I switch off?